It's my great pleasure to introduce Jian Zhou. So Jian's really been a leader in the field of using deep learning to represent sequence to function relationships, kind of from the beginning of this becoming like a mainstream area in computational biology. So did really like groundbreaking work with Olga Troyanskaya, developing DeepSea, one of the early applications of convolutional neural networks to predict transcription factor binding, represented by ChIP-seq, and lots of kind of really great work in that sort of direction. So predicting RBP binding, which is an area close to my heart as well. And then in his group at UT Southwestern, they've been doing really great work looking at these really challenging tasks like predicting chromatin conformation as represented by Hi-C, with this ORCA method, some really amazing engineering efforts there, and most recently understanding transcriptional regulation using interpretable deep learning models, which I think is what you'll be talking about today. So excited for your talk, and thank you for coming.
Yeah, thanks, David, for the kind introduction. And I would like to thank the organizers for the invitation. Today I will share with you a story about discovering how the sequence controls the transcription initiation. So to start, I will start with a probably somewhat provocative statement, right? So genomic sequence might be the most underexploited resource in genomics, right? So what do I mean here, right? So of course, that genomic sequence is being used every day in all kinds of research. However, I know with the exception of many of you here, but most of the time, when genomic sequences were used, it's often used as a way to make a measurement, right? So we use the reference genome to do all kinds of measurement about the genome, and it's used to detect mutations. But the actual sequence that's encoded in the genomic sequence, right, the biology that's encoded in the sequence, is still relatively rare to be extracted as a way to drive those biological discoveries, right? So most of the discoveries were not made from extracting this information from the sequence. So compared to how much biology is already encoded in our genome, so there's a huge gap between that and how much that we can use that for discoveries, right? So I think there is a huge gap here, and also like a great opportunity to, by reading more information out of the genome and use that to help us gain better understanding of the biology. Right? And to bridge that gap, of course, I think the challenge is that it's still hard to understand the genomic sequence, right? So I think understanding the genomic sequence is a problem that is, it's quite hard for traditional approach, but it's almost perfectly set up for machine learning approaches, right? So reason for that is that first we know, of course, our entire genomic sequence in the human genome, the 3 billion base pair. And at the same time, we also have tens of thousands of different genomic data sets that's measuring various kinds of activities about the genome, right? Let's put some random ones here, from the transcription factor binding to the 3D genome organization to the transcription in increasingly more cell types, right? However, just having those genomic data, it won't directly tell us about how are those activities determined, or at least depend on the genomic sequence, right? So those are the information that we cannot directly measure, but we can infer through a modeling approach, right, by building machine learning models, right? So in the space of capturing the dependencies between the genomic sequence and its activities, I think of it as 3 main directions that we explore. So the first direction is, of course, prediction. So we want to go from the genomic sequence to predict its activities, right? So it's also where I started from, and in the past, we've done a lot of work in developing sequence-based models to predict various kinds of genomic regulatory activities, right? And as we build those models, we think they are great tools for us to understand the underlying mechanisms of genome regulation. But I also gradually realized that just having a predicting model, so being able to predict something does not equal to being able to understand that, right? So a lot of time, I think we certainly get the feeling that our model understands it. It can certainly understand something that we haven't understood about the underlying biology. But we don't know what it is, right? So I think it is still a non-trivial task to be able to extract that understanding from the predicting models. So it is a challenging task on its own, and also I think it requires development of methods specifically for understanding on its own. So I put it as a separate direction here. It's also one direction that I will be mostly focusing on today. And the third direction is kind of the flip side of prediction, right? So instead of being able to go from the sequence to predict its activities, we also want to be able to do it the other way around. So we want certain kind of regulatory activities. Can we design sequences that give us that activity? So I think there is a synergizing effect among all three of those directions. So today I will be focusing on a study of transcription, and more specifically transcription initiation. And I'll be mostly touching upon the direction of prediction and understanding, but it's also one direction that we have done a bit of work on all three of those directions. So we started from asking a simple question. So by the way, this is work with my brilliant graduate student, Sinia. And so we started by asking this question of so what is a promoter, right? So what defines a promoter at the sequence level? So of course, we know that promoters are essential part of a gene, right? So you can have a gene that does not encode a protein, or you can have a gene that does not have an intron, but all genes have to have promoter to be transcribed, right? So it is one of the most essential elements in the genome. But perhaps surprisingly, we think of what should a promoter look like at the sequence level in the human genome. So our understanding to that was still quite incomplete. So in the past few decades, there have been quite a bunch of those called promoter motifs have been identified from many different organisms, right? But if we look at the human promoter, so even a strong promoter, it might have none of those motifs, right? So like the Tata motif, depending on our threshold, we think it's present in no more than 10% of the human promoters. So for most of the human promoter, you look at the sequence, it's still hard to tell why is it a promoter and why transcription starts at the exact position that it starts at, right? So it's still something that we didn't have a full understanding about. So to tackle this question, we want to just first formulate it as a machine learning problem, right? So what we want to do is that we want to build a model that can start from the genomic sequence and predict the transcription initiation activities at the base pair resolution. So those are one examples of the transcription initiation site measurement by phantom cage. So those measurements give you the precise 5' end of the transcript. So you can count how many transcript start sites are detected for each base pair position. Right? So you plot that on the axis of genomic positions, you can obtain a plot like this. So the y-axis is on the log scale. So you see this wiggling of those activities across different positions, right? So those wigglings are not noise. So it's actually highly reproducible across samples, and even reproducible across cell types, right? So we think those curves, those wiggling shape here, actually reflects the underlying mechanism of the transcription initiation at this promoter, right? So if we are able to capture how are those shapes determined at the sequence level, we might have captured something about the mechanism of the transcription initiation, right? So we first built a deep learning model to be able to predict that from a 100 kilo base pair of sequence. And also, it turned out that we are not the only one who has this idea. So Onsho's lab at Stanford, also Charles Dunkel lab at Cornell, also independently develops those methods that does similar kind of prediction. And you should also check out those work. And I think the author of ClipNet, Adam, is also here in the conference, so you should talk to him as well. So what we did a little differently in our approach is that we are able to just study this deep learning model that makes this prediction very well and summarize what we learned from the model and use it to develop a simple model that can also do a very good prediction of the shape of the transcription initiation, but only using 600 base pairs of input and also a very small amount of parameters. And we can interpret pretty much every one of those parameters. Right? So this small model that we call puffing is the focus of this talk. So before I go into how does this model work, just show you the prediction performance of this model. So this example of a promoter and its transcription initiation activity profile predicted from the simple model, the deep model, and the experiment. Right? And just two examples of that. And of course, we can quantify this performance by looking at the correlations of the model prediction on a holdout chromosome that the model has never been trained on. Right? So we summarize those correlations. We see that for the most expressed promoters, where we have the most measurement, the prediction correlation can get to up to 0.9, right? And with the promoter that has lower transcriptional activities, the correlation is lower, but it's also partially due to the noise in the measurement because we look at the correlations between two different experimental measurements of phantom cage and encode cage. We also see a decrease in correlation with the lower activity promoters, right? So overall, the puffing, even though it's not as good as a deep model, it is still doing pretty well in capturing the shape of the transcription initiation. And also, both of them are actually able to achieve higher correlation compared to when you compare the two different but most correlated experimental techniques, right? So which says that the model should have captured quite a lot about what determines the transcription initiation. So even though it is a very simple model. Right? So how does this model work? So we summarize it in this diagram. And actually, it's very simple, just doing 2 steps of computation, mainly. So I just go through that step by step. So the first step is that we start from the genomic sequence, and we scan for motifs that are present in the sequence, right? So how it's done is through there's a convolution layer with the soft flux activation function, and you get this motif activation profile. So we use different color to indicate different motifs, right? Just detecting the motif positions. And next, the second step, the only other step of computation that's needed is that we convert these individual motif activations into its corresponding effect, like this red curve, which is Tata activation is converted to this red effect, right? And this NFY motif activation is converted to this curve of NFY effect. So that is, this effects are applied through another convolution layer. And then we only have to sum up the effects at each individual location. And surprisingly, that is sufficient to make a good prediction for most of the promoters, right? So what I didn't tell you is that, so in addition to the motifs, there are also 2 other simpler types of sequence patterns that we detect, which effects were also added to the motif effect to obtain the final prediction. So the 2 other sequence patterns are initiators, which are only affecting very local ±7 base pair window to fine-tune the base pair resolution transcription initiation signal. And the other one is a trinucleotide, which is just the residual dependency that we couldn't explain, but we can capture that with just a 3-nucleotide effect. So as we sum up all these sequence pattern effect per position, it gives us our final prediction already. So which is, you can interpret it as the log of the transcription activities, and we just need to convert it into the log +1 scale to compare with the experiment. Right? So I would like to also show you that we can have this model working live in a web server that we made. Right? So this is you go to like puffin.jolab.io. So this is a server that actually loaded up this model that you can look at, not just look at the sequence, and you can also edit them. Right? So if I remove the Tata, you see the prediction decrease. Right? And if I can remove the other motif, the prediction will decrease even further. Right? So this is a way that you can actually just interact with the model live on the website. And you click on demos, it shows you the predictions for many different promoters. Right? So I'm just showing this to give you a sense of how diverse these promoters are. Right? So almost every one of them have distinct combinations of motifs. But following the simple rule, we can predict quite well about the transcription initiation activity in all of those promoters. Right? So going back to the model, so how does it make this prediction? Right? So the core of it are the motifs that it has learned from this task of predicting from sequence to the transcription initiation activities. Right? So all of those motifs were learned from scratch. We didn't give it the motif. But after we have found those motifs that has been repeatedly learned as we train multiple rounds of those models, we figure that most of them actually match known motif. Right? So that's why we can give it the names here. But with the exception of Tata, actually for most of those motifs, we were not known. It's not known previously, like whether and how it contribute to transcription initiation, right? Which was captured in the motif effects that are learned here, right? So those are probably the most key information that has been captured by the model, right? So how to read this graph is that if we position the motif at the center, we place the Tata at the center here, it will activate transcription at almost exactly 30 base pair downstream of that motif, right? So if you put the YR1 motif at the center, it's going to activate the transcription at almost immediate upstream of that motif, and also has a weaker effect, about 200 base pair upstream of that. Right? So we can also resolve the effects on the two different strands. Right? So as you can see here, both Tata and YR1 has pretty strand-specific effect. Right? So it has different effects on the forward strand transcription and the reverse complement transcription. Right? But there are also a group of motifs which have almost symmetric effects on both strands. So it activates transcription on both strands simultaneously with almost identical effects. So we call those bidirectional motifs. Right? So those bidirectional motifs, they seem to share some similarities in their effect curve. But still, each one of them has a distinct shape. Like for example, for NFY, you see this about 10.5 base pair periodicity, which matches with the helical periodicity of DNA, which rotates every 10.5 base pair to the origin. Right? So we think that reflects that NFY might have a more rigid interaction with the POL2 pre-initiation complex. Right? So it has to bind at a certain surface relative to the POL2 pre-initiation complex. Right? So perhaps each one of those curves are actually reflecting the underlying molecular mechanism of how does those motif and the transcription factor activate transcription initiation. Right? So There's a special motif here that we included, but we know that it does not affect transcription initiation, or have very little effect on transcription initiation. So the reason that we can know this is that we built models for different types of experimental techniques. So including those that measure mature transcript, like CAGE and Rampage, and also those that measure only nascent transcript, like ProCap and GrowCap. So this motif, the UNSNRMP, also known as the 5' splice site, it only has this effect when we are looking at the mature transcript, but it has no effect if we are looking at nascent transcript level. So, which means that it likely affects the amount of transcript after transcription initiation happened, right? Which is also consistent with recent studies which show that it has an effect in promoting the transcription elongation. And finally, there was one motif that are highly directional, and initially we didn't recognize it. But later we realized that it is this very long motif. It's actually a combination of several previously identified co-promoter motifs, including the initiator, which is where the transcription starts at, and the several downstream motifs here. Right? So all of those motifs were considered to reflect the preference of TF2D, part of the pre-initiation complex, and I also think that this long initiator might be reflecting the overall preference of TF2D. Right? So with those effects, so now we also want to be able to just more directly test or verify the effects that the model have captured. Just beyond the fact that these motifs are reproducible and able to make good predictions on the whole chromosome. Right? So one thing this model can allow us to do, because it is a simple and interpreted model, which we know that how each element in the model do, is that we can now predict the effect of removing a transcription factor from the system. Right? Because we know which kernel corresponds to NFY motif activation and motif effect, we can just remove that NFY activation and see what are the predictions of the transcription initiation signal after we have removed NFY. Right? So because NFY will activate transcription downstream and slightly repress transcription upstream, if we remove them, generally the effect is a decrease in the downstream and a slight increase in the upstream. Right? So we can summarize the prediction here. Right? So depending on the NFY position and all the other motifs in the promoter, we see different effects at different promoters. Right? So here it is sorted by the activation effect to be able to more easily see the trend. And there is a data set that has been generated that did an NFI knockdown. Right? So we compare with that data set and show that experimentally we can see a very similar pattern as predicted by the model, right? Which is the repression downstream and activation upstream. And we're also able to test that for another motif that we predicted to have an effect on transcription initiation, which is YY1. Right? So this is a pretty effect of YY1 knockout, and this is the experimental effect of depleting YY1. Right? So we also see that the predicted effects were indeed observed in the experiment, and even the secondary, like the less strong effect of YY1, about 200 base pair upstream, can also be observed in the experimental measurement. Right? So this effect was actually not originally reported in this paper, but when we digging into the data set, we found that actually there is an effect of YY1 knockdown, YY1 depletion on the transcription initiation. Right? So we can also test it in the waves of mutating motifs. Right? I'm not going to go too deep into this, but just show that the model can also reproduce the experimental effect of inserting and deleting those transcription factor motifs. And after our preprint is published, there have also been a couple of other studies that have been consistent with our prediction. So for example, one of the studies that tried to look into the ZNF143, which is actually one of the surprising factors that we were not expecting when we were building the model. They were looking into it with the expectation that it might be a looping factor, but it ended up finding that it is a transcriptional regulator, which can, if you mutate it, it's going to significantly decrease the transcription signal. Right? So another study that also verified that those motifs that we also captured in the model will have an activating effect on the downstream of this motif and the repressive effect if it is the transcription start site is on the upstream of those motifs. Right? So beyond those experimental validations, we can also look at the evolutionary conservation. And somewhat to my surprise, you can also see this trend quite clearly just by looking at the conservation signal. So what is showing here is that if we look at the conservation of motifs, it actually depends on where it locates relative to the transcription start site. So the motif will be more conserved across species if it is located at the position where it has the maximal effect on the transcription start site. Right? So like Tata has a maximum conservation when it is located about 30 base pair upstream, and Y1 has a maximum conservation when it is just downstream of transcription initial start site. And even this periodicity of NFY can be seen through looking at the evolutionary conservation and analyze its dependency on the distance to the transcription start site. Right? So all of those are supporting what we have learned from the model. So now we're quite confident about what the model has learned about this, how does those motifs contribute to transcription initiation. And now we want to ask, like, what does it mean for the gene, right? If we know which motifs are contributing to the promoter, right? So we can now summarize the contribution of each motif to every promoter into a score, right? So here it is an example of the contribution scores for all of those motifs to top 100 most expressed promoters in the human genome. Right? So in the highest expression promoters, you see an overrepresentation of Tata motif, but still the contribution is quite diverse. Right? You see many different combinations of those motifs, which is generally what we see for those promoters. We can do this analysis for all the top 40,000 promoters, and we just sorted by the motif that has the maximum contribution to this promoter. So SPs are the motifs that contribute to the most promoters, but each one of these co-promoter motifs are contributing, is the dominant motif for a fraction of these promoters. Right? And overall, we see when we look at the correlation between the any pair of motifs, it's never very strong. It's always below 0.1 correlation. So we really think that you can very flexibly combine those motifs to make a promoter. Right? Yeah. But what does those motif contribution mean for the gene? Right? So all of those motif bind transcription factors that are ubiquitously expressed, even though the expression level can be a little varying. So I don't think any one of them will have a strong effect preference to a certain tissue, even though ETS is slightly have slightly higher effect in blood lineage instead, especially T cells. Most of them are having similar effects across tissues. However, we do see a pretty strong correlation between the motif contribution and the expression variation across cell types. So here we're looking at the expression level and its dispersion across cell types. So each dot here represents a gene, and the x-axis represents the expression level of that gene, and the y-axis represents the dispersion of that gene. So I should say promoter, right? So it's actually quantified at the promoter level. And the color here indicates the contribution of Tata motif to this promoter, right? So a clear trend that you can see here is that with higher contribution from Tata, the promoter is more likely to be variable across cell types, right? Even at the same expression level. And in contrast, if the promoter has very high YL1 contribution, it tends to be more ubiquitously expressed and less variable across cell type. So all of them, each one of these motifs actually has its own preference in this spectrum of preferring this more tissue-specific expression pattern versus the more ubiquitous expression pattern. And the U1 SNRMP, the 5' splice site, as we mentioned, that it should not affect the transcription initiation. It's kind of neutral in its preference to the tissue-specific versus ubiquitous expression pattern. So why is that? So why are those motifs preferring different tissue specificity level? So we have a hypothesis about that. So I just first tell you what's the hypothesis, and then show you why we think that is the way. So the hypothesis is that the motif composition will tune the responsiveness of those promoters to the transcription regulatory signals like enhancers. So the Tata promoters might have a higher dynamic range than the Y1 promoters, which may have a lower dynamic range and less responsive to the regulatory signals. So the reason we think of it that way is also because we look at the promoter activities in an almost orthogonal way, and we see a similar order of motifs as the tissue specificity. So here, we just insert a lot of promoter sequences into different genomic locations and test, and we predict its activity from the deep learning model, right? So what we found is that if we insert a promoter which have a strong Tata contribution, then this promoter is more selective. It is expressed in fewer positions relative to the max expression level, whereas Y1 promoters are less selective. It is more likely to be expressed in more positions where we insert it into. And this general order of motifs is quite consistent with what we see in terms of the tissue specificity. So it's still a hypothesis, but we think that that might be what explains the preference of those motifs for different levels of tissue specificity. And finally, what we can explain with this model, you might notice from introducing those effect curves and bidirectional motifs, is that why those most human promoters we see transcription initiation on both strands, on both directions, if we look at it at the nascent transcript level, right? So most of the human promoters are like that. If you look at the nascent transcript, you most likely see transcription on both directions, right? If you look at the mature transcript, it is much more unidirectional, right? So we think that bidirectional transcription is due to the bidirectional motifs which activate the transcription initiation on both strands, right? And for the mature transcript, you have more contribution from factors like the 5' splice site, which only affect the transcription, mature transcript level on one strand, right, which leads to more directional output of the transcript. So we can also do this quantitatively. So you can also quantitatively analyze what are the contribution to each strand at base pair resolution. So here is an example where this is an experimental measurement, and it's our prediction. And we can quantify how much each base pair contribute to the forward strand, and how much each base pair contribute to the reverse strand, and compare how much they are shared, right? So this is a contribution to the forward strand. We can also decompose it into contribution through different motifs, right? And this is the contribution to the reverse strand. And as you can see that it is very similar to the forward strand. And we summarize this for all of those forward and reverse transcription star site pairs, we find that if the distance is smaller than 300 base pairs, more than 90% of those contribution scores has more than 0.75 correlation, which means that they're quite shared. They have both the forward and reverse strand transcription has very similar sequence contribution. So finally, we also want to know how conserved are these, what's the model captured across different species, right? So we can train this model for both human and mouse because both of them have very similar experiment done through the Phantom Kit project. So we can train separate models on human and mouse data and make predictions on the other species, right? So to summarize, what we see is that the model that's trained on human and mouse are basically giving us identical predictions. So the correlation is very high, very close to 1 when we compare it. And moreover, if we compare the human model and mouse model prediction, and we only evaluate on the mouse experiment, actually you don't see a human model having a lower performance compared to the model that is trained on mouse data, right? So I think it's really that it is almost identical in terms of how does transcription initiation depend on the sequence between human and mouse. And we also want to look more diversely across mammalian genomes, right? Which is hard because we don't have those transcription initiation measurement for all those other species, right? So we did what we can do. So we try to look at the evolutionary conservation. So previously, I've shown you that if we look at it from the human genome perspective, the motifs are more conserved when it is located at position where it has stronger contribution to transcription star site. So now we do it from the viewpoint of each of the 240 mammalian genomes, right? So if you are just starting from each one of those genomes, you do have to take assumption that the transcription star site is conserved from the human to that species. With that assumption, you see that, so here actually there are 240 curves here, each represent the conservation in one species. They all share a very similar trend of the position dependency, right? Which seems to suggest that it is likely that this sequence dependencies are also conserved across mammalian genomes. So to summarize, that we are able to build a simple model that can predict from the sequence to the transcription initiation signals. And this has been able to allow us to understand something about the promoters, including like what are the sequence dependencies and how does those motifs contribute to the promoters. And we are able to use that to stratify promoters by motif contribution and found that certain motifs have different preference for tissue specificity. And we can use that to explain the directionality of those promoters. And we can look at the conservations across different species. And just finally, I think to look ahead, I want to just revisit this okay, triangles that I posed at the beginning, right? I think here, I think mostly just looking at the synergies between the predicting models and the understanding, right? So we're looking at how predicting model can help us gain a better understanding about the underlying process. I think there are still a lot more synergies here that has not been explored. For example, if we gain a better understanding, can we use that to design better predictive models, right? So can we use that to design new promoter sequences? And if we design some sequences and we measure its activities, can we use that to verify or refine our understanding, right? And I think there's really a lot of synergy between all three of those directions, which can hopefully improve our ability to do each one of those tasks. And hopefully, I think it can help us get to the question that I posed at the beginning, so we can use that to extract more of the biology through studying the genomic sequence. So yeah, finally, I'd like to thank my group who have contributed to this study, which is Sinia, who is the first author, who works with me mostly on the computational side, and the Donghong and Jian Zhou from St. Jude did the experimental validations of this study, and Chen Aishi developed the website. So thank you for listening, and I'm happy to take questions.
Yeah, we definitely have time for some questions.
Hey, so I have a technical and a conceptual question. So for the motifs that appear symmetric in terms of activation, they're not exactly the same.
Right.
Do you know if that's like up to statistical error or there's something meaningful there? And my second question is, how much do you have to worry about dynamics, right? Because some RNAs are initiated, but they're immediately degraded, and then you don't observe them.
The first question, I think, is from the reproducibility, we do know that this slight asymmetry from the forward and reverse strand are actually reproducible. So if we train the model multiple times, we consistently see these differences. So I do think there is some slight asymmetry, even though that is almost symmetric. And for the question of the dynamics, so I'm not sure I completely get your question. So there might be some of the intermediate transcripts that were not able to be detected through the existing technique. Is that?
It's not like a flaw in your approach. I was just wondering if you've thought about this, that in theory there's a bunch of transcripts that are initiated, but they're degraded, and you don't observe them even in the cage data, and like how that factors into how you think about the process.
Right. I do think that the current model is kind of, as you said, I think kind of a static way. I think the way that I understand the Puffin model is that we have those motifs, and each of them have can probably alter the binding energy of the pre-initiation complex, right? And that sum up to the energy profile of the pre-initiation complex binding at every position, which leads to the transcription, right? But I think it is. This is probably not the full picture still, right? So it's a first order approximation, probably not, there's still probably a lot of other things that are missing. And one thing that I do now is try to think about is like how does a model these dynamic processes like happening in the genome? Like how does the order of things affect the outcome, right? So if the transcription factor binds first and another transcription factor binds second, or like if there is some order of transcription, minor burst of transcription event, and it might affect the later transcription level, right? So how would those dynamics affect how we should be building our models? I just don't have an answer to that, but I think it's certainly a question that we should be thinking about in terms of building those interpretable models. Yeah.
Hi, beautiful talk. I wonder how it would look like if you distill all this to a set of rules, basically describing what makes a promoter. Is it simply 8 or 9 exclusive groups of promoter classes, or can you go more granular and define in terms of different combinations?
I mean, use that to group the promoters into different classes, right? So I think first of all, what we tried here is to look at the promoters sort of classes based on which motif contributes the most to that promoters. Which does show different properties of those promoters by the most contributing motif. But on the other hand, do I really think that these promoters are well clustered into different clusters? I think not necessarily, because we only look at the correlations between different motifs. The contribution and correlation are usually pretty low. So there is a quite flexible way of combining those different motifs into different promoters. So I think it's more of a continuum rather than having different classes, even though having different motif contribution will likely still affect the property of those motifs, of those promoters.
Thank you for the great talk. So I think in your one of the last slides, you were showing that the multiple models that you have developed over the time, and for example, I think there was Expecto and Puffin, I think grouped together. So when these different models are in the literature, and there are like different groups like developing these models, do you think like we should come up with on consensus on the folds to train them on, validate them on, and test them on? For example, for expecto, I think you proposed removing the paralogous regions from the test set. So, and did you follow the same procedure for puffin test sets to remove probably the paralogous genes or the homologous promoters? Thank you.
Yeah, I think that's a very practical question, I think, for those of us who develop those sequence models, right? I think it would be very helpful if we can all compare with each other's models on the same holdout set, right? So, our models, we do always try to be consistent in the holdout sets so that it's comparable across all the models that we develop. But certainly, I think different groups can use different holdouts. So if they are holding out different chromosomes and different regions, then it is harder to find just the intersection to evaluate the models. I think it will be indeed very helpful if I think the field can agree on a certain way of doing the holdout, or as David Kelly's group have been recently doing, that you actually train models using just many folds, many cross-validation folds, so that you can actually have different models that are holding out all parts of the genome, right, in different models, so that also allows people to compare with each other more easily. So I think both of those will be very helpful for the development of models in the field.
Great talk, Jian, and very interesting study. It's very interesting that some of the motifs like Tada warnings has more cell type specific like driving ability in promoters. I wonder, have you thought about, let's say, design a synthetic promoter that, well, usually we think that the cell type specificity or tissue specificity came from enhancers. Have you thought about design a specific promoter that can boost the expression of enhancers and maintain the specificity of those enhancers?
Yeah, I think that's certainly something that we are interested in exploring. So following what we think, I think we build a strong Tada promoter that it should be more responsive to enhancers compared to we build a YYM promoters, right? But how do we balance the expression level versus specificity? I think that is still something that we don't know, and I think it will be a great question to ask in the future.
I think, unfortunately for time, we probably need to wrap up there. Let's thank Jian again for a great talk.